Today, we are diving into some absolutely incredible new research that could totally change how we fight cancer. We're talking about turning our own immune system into a hyper-targeted weapon, not in some far-off lab, but directly inside our bodies. So let's get into how this groundbreaking technology actually works. Just think about that for a second. What if, instead of a long, complicated manufacturing process, a simple injection could command your own cells to become elite cancer killers? Well, that's not science fiction anymore. It's the exact premise of the technology we're breaking down today. All right, here's our roadmap for this explainer. First, we'll look at an amazing but kind of flawed cancer therapy we have today. Then, we'll explore this radical new idea of making the medicine inside the body. We'll break down the brilliant three-part tech that makes it all possible, look at the jaw-dropping results, and finally, talk about what this could mean for the future of medicine. Okay, so to really get why this new approach is such a massive deal, we first need to talk about one of the most powerful tools we currently have against certain cancers, something called CAR T-cell therapy. You know, the concept here is just revolutionary. Doctors take your own immune cells, specifically your T cells, and they genetically engineer them in a lab so they can recognize and obliterate cancer. They literally create a living drug that is incredibly effective for some blood cancers. But, and this is a really big but, the process is far, far from perfect. So the current method, which is called ex vivo, meaning outside the body, is just incredibly challenging. Every single treatment is personalized, and it can take 4 to 7 weeks from start to finish. That's a crazy long, anxious wait for patients who are already very sick. And on top of that, it can cause some really severe side effects and even carries a risk of causing other cancers, which is why the FDA requires a scary black box warning. And this is where the breakthrough at the heart of this paper comes in. What if you could skip the whole lab part entirely? What if you could do that same genetic engineering in vivo, directly inside a patient's bloodstream? This image here basically shows you the entire core idea. Tiny little nanoparticles are infused into the blood. They're designed to find the exact right T cells, deliver a set of genetic instructions, and boom, the T cell is instantly reprogrammed into a cancer-hunting CAR T cell. The body itself becomes the factory. So, how does this microscopic magic trick actually work? Well, it all comes down to this brilliantly engineered 3-part delivery system. Let's break it down. All right, this diagram lays out all the key pieces of the puzzle. At the very center of it all is the lipid nanoparticle, or LNP. You can think of it like a delivery vehicle, but honestly, it's more like a super high-tech microscopic drone with a very, very specific mission. Let's just stick with that delivery drone analogy for a second. First, you've got the vehicle itself. That's the LNP shell, which protects its precious cargo. Second, you have the targeting system. This is a special molecule called a nanobody, and it basically acts like a GPS or a postal code, making sure the drone only delivers to one specific address, the CD8 plus T cell. And third, you have the payload, the messenger RNA, or mRNA, which carries the instructions to build the cancer-destroying weapon. And using mRNA here, that is an absolutely critical choice. See, unlike traditional gene therapy that permanently changes a cell's DNA, mRNA is just a temporary message. It tells the T cell to start making the cancer-fighting receptor for a few days, and then it just disappears. This is a huge safety advantage. It makes the therapy controllable, and you can even redose it, all without the risk of permanent genetic changes. Okay, so the design is elegant, it's brilliant, but the only question that truly matters in medicine is, does it actually work? Let's look at the data. First up, in the controlled environment of a petri dish, the answer was a huge, resounding yes. The LNPs successfully reprogrammed the T cells, and those brand new CAR T cells were incredibly precise. They killed the target cancer cells while completely ignoring healthy cells or any other cells that weren't their target. But of course, that's the million-dollar question, isn't it? It's one thing for it to work in a lab dish. It's a massive leap to go from that to the chaotic environment of a living organism that's battling an aggressive blood cancer. And the answer is, well, just look at this. It is absolutely stunning. On the left, you see the control groups. Those bright, glowing areas show aggressive tumors spreading like wildfire. But on the right, in the mice that got the in vivo CAR T therapy, the tumors are almost completely gone. The visual evidence here is just undeniable. And this chart here, it just puts numbers to what we just saw with our own eyes. The 2 control groups, their tumors just grew exponentially. But that blue line, representing the group that got the targeted LNPs, it shows a dramatic and sustained suppression of the tumor. It works, and it works powerfully.
So the science is incredible. We've established that. But what does this actually mean for patients? Why is this a potential game changer for the entire field of medicine?
This slide just lays it all out, the old way versus the new way. You're potentially replacing a weeks-long, highly complex process with an off-the-shelf therapy that can be given immediately with a simple IV infusion. This research suggests it could even get rid of the need for the harsh chemotherapy patients currently have to go through. And you're swapping a permanent, potentially risky genetic change for a much safer, temporary, and repeatable one. And hey, you don't have to take my word for it. The researchers themselves don't mince words. They see this as a platform that has the promise to become a true off-the-shelf treatment, which could democratize this kind of therapy and make it accessible to countless more patients all over the world. And that brings us to the final, really tantalizing thought here. This isn't just about a better way to treat cancer. This is a platform technology. If we can safely and effectively reprogram our own immune cells inside our bodies to fight cancer, what else could we teach them to do? Autoimmune diseases? Fibrosis? The possibilities are just extraordinary.